Good day, this is Professor Will, CBMD PhD. Today's Wednesday, November 20th, 2019 at 6:28 seconds Eastern Standard Time. And this is a reading on the functions of cytoplasmic membrane. So let's get started here. The cytoplasmic membrane of bacteria is a very busy site, at which an almost bewildering number of important processes occur. It carries out most of the reactions that are handled by the many organelles of eukaryotic cells. Since bacteria are generally far more metabolically capable and diverse than eukaryotic cells, it is not surprising that an estimated 1/4 of the cell's proteins are membrane-associated. One of the key principles in biology is the basic universality of bioenergetics, which refers to the process of energy generation, storage, and utilization in cells. The major feature of these processes is the use of ion gradients that are formed across cellular membranes, such as the membranes of mitochondria and chloroplast in higher organisms or the cytoplasmic membrane of bacteria. The major feature of these processes is the use of ion gradients that are formed across cellular membranes, such as the membranes of mitochondria and chloroplast in higher organisms, or the cytoplasmic membrane of bacteria. In most systems, the gradient of protons is an essential factor in bioenergetics, although gradients of sodium ions are used by some bacteria for energy generation and by most eukaryotic cells for cellular signaling and nutrient transport. Ion gradients are the intermediate between the processes of electron transport, and the formation of ATP has been overwhelmingly accepted. In bacteria, ion gradients are also used to drive several types of active transport systems: bacterial motility and protein secretion. Energy generation refers to the trapping in a metabolically useful form of the energy absorbed from sunlight, or released by reduction of some inorganic molecule, or the oxidation or breakdown of organic molecules serving as energy source. Bacteria can generate energy by many different processes, in simple fermentative pathways such as the glycolytic breakdown of glucose to lactate. ATP can be formed during several enzymatic steps by the process of substrate-level phosphorylation. Important energy-producing processes use electron transport chains to pass electrons from a carrier of high negative redox potential to carriers of successfully lower energy state. During respiration, electrons enter these chains following transfer from an organic molecule and are ultimately transferred to an inorganic electron acceptor. During photosynthesis, electrons are excited to a higher energy state following absorption of light by a chlorophyll-related molecule. Summarizes several key steps of microbial energy generation. During the process of electron transport to carriers of successfully higher redox potential, there is a separation of charge across the membrane. This is ultimately coupled to the movement of protons from one side of the membrane to the other, resulting in the formation of a proton motive force, PMF, which has two aspects. Movement of the positively charged proton creates an electrical charge across the membrane. In bacteria and mitochondria, in which electron transport results in the release of protons, the electrical charge is negative inside and can be 100 to 200 MV. Proton pumping also results in a difference in proton concentration, or delta pH, across the membrane, such that the exterior is usually more acidic than the interior. Photosynthetic or respiratory electron transport thus results in proton pumping and creation of the PMF. The proton gradient can be tapped to bring about the formation of ATP, which is the ultimate energy source for most energy-requiring processes in the cytoplasm. Synthesis of the ATP is carried out by a family of protein complexes, which include the F1 and F0 protein translocating ATPases of mitochondria and bacteria. Related complexes are found in chloroplast and archaea. These protein complexes contain a membrane-embedded sector, the F0 portion, which includes the pathway to allow the protons to flow back into the cell in response to the chemical and electrical forces acting on them. The F1 portion of the complex contains the sites for conversion of ADP plus pi to ATP, and the energy for this process is coupled to the movement of protons. The stoichiometry of the process is such that the entry of 3 or 4 protons results in the formation of the molecule of ATP. The individual steps of proton movement through F1, F0, ATPase, and ATP synthesis or hydrolysis are tightly coupled under most conditions to prevent wasteful loss of the PMF or of the ATP pool in the cell and consequent heat generation. In bacteria, this ATP synthase functions in a reversible manner to allow ATP that was generated by substrate-level phosphorylation to drive formation of a protein gradient, which can then be used to drive transport systems or motility. Movement of protons causes the F sub-0 sector to rotate within the membrane, like the action of a turbine. The rotation of the F sub-0 sector is coupled to changes in the conformation of the nucleotide binding sites, the stationary F sub-1 sector, which is linked to the interconversion of ADP plus PI to ATP. Photosynthesis traps energy of sunlight and converts it into metabolically useful forms of ATP during cyclic electron transport and NADPH during non-cyclic electron transport processes. In some organisms, photosynthetic electron transport is coupled to the formation of oxygen from water, a process that is essential for aerobic life. There are numerous pigments in cells that absorb light energy for use in photosynthesis, but the most important of these are the chlorophyll. Chlorophylls are porphyrin molecules similar to heme, but containing a magnesium atom instead of iron. Some pigments are carried in the protein molecules that serve as an antenna or light harvesting complexes. But the key processes occur in a protein complex called the photosynthetic reaction center, which typically consists of 3 proteins that spread across the membrane and contain bacteria chlorophyll, bacterial phenophytin, and menaquinone, and non-heme iron as electron carrier. The light energy is ultimately absorbed by a chlorophyll molecule, and this energy excites an electron to a higher energy level. This excites electron passes through a series of electron-carrying prosthetic groups within the reaction center and then out of the pool of membrane-bound quinones, which transfer the electron to the cytochrome containing electron transport chain. The passage of the electron transport through the electron transport chain is coupled to a pumping of protons across the specialized membranes containing the photosynthetic apparatus. In cyclic phosphorylation is carried out in the photosynthetic bacteria, the electron ultimately returns to the chlorophyll molecule after transfer to the paraplasmic heme protein cytochrome C. In the process of non-cyclic phosphorylation in plants and cyanobacteria, the electron can be transferred ultimately to pyridine nucleotides for the use of reductant and biosynthetic processes. In this process, the electron can be replaced by chlorophyll, by another light absorption process that removes electrons from water to create oxygen. A completely different system for conversion of light into metabolic energy is present in Halobacterium salarum, an extremely halophilic archaean that thrives in very saline environments such as Dead Sea or brine evaporation pond. The consequence of these changes is that the protein is released from the bacteriodopsin on the outside of the membrane and replaced by one from the cytoplasm. In this way, light is directly converted into a transmembrane protein gradient without the requirement of an electron transport chain. Respiration is a process whereby electrons from the metabolism of an energy source are transferred through a protein pumping electron transport chain to some inorganic molecule. The most familiar form of respiration is aerobic respiration, in which oxygen serves as the ultimate electron acceptor. Owing to the ability of oxygen to accept electrons, aerobic respiration is the most energetically favorable but some partially reduced forms of oxygen. Hydrogen peroxide, superoxide anion, and hydroxyl radical are extremely reactive and thus toxic to the organism. Many bacteria are capable of carrying out anaerobic respiration, in which the electrons are transferred to alternative acceptors such as nitrate, nitrate sulfate, or sulfite. These processes yield less energy but can occur in anoxic environments. All respiratory metabolism uses electron transport chains, whereby electron transfer from the donor to oxygen or other acceptor is coupled to protein movement across the membrane. In E. coli, electron donors for respiration include NADH, succinate, glycerol, 3P-formate, lactate, pyruvate, hydrogen, and glucose. Electron acceptors include oxygen, nitrate, nitrite, fumarate, dimethyl sulfoxide, trimethylamine, and oxide. Typical respiratory systems contain 2 to 4 transmembrane protein complexes. These include substrate-specific dehydrogenases, which transfer electrons from the donor to quinones in the membrane. The reduced quinones migrate to another protein complex which accepts electrons from them and transfers the electrons to cytochromes and ultimately to the terminal electron acceptor. The transmembrane protein complexes often contain flavin and or non-heme iron and are arranged in the membrane in such a way that the passage of electron results in the release of proton to the outside or its consumption from the cytoplasm, i.e. the formation of the PMF. Coupled processes. Some bacteria couple the transport and metabolism of their energy source directly to the production of the PMF. An example of this very simple but not very energy-rich process is malolactic fermentation in Lewis-Nostock. The substrate malate is transported into the cell and converted into lactate, which leaves the cell in exchange for a new molecule of malate. The net result of this process is the movement of one negative change into the cell and the consumption of one proton inside the cell, which results in the creation of a PMF that is interior negative and alkaline. Membrane transport. Biological membranes form the permeability barrier separating the cell from its environment. The hydrophobic barrier of the membrane biolayer greatly restricts passage of the polar molecule, although nonpolar molecules can pass. Transport mechanisms exist to move nutrients and precursors into the cell and metabolic products, surface components, and toxic materials of the cell. Several types of transport mechanisms and families of transporters have been identified. Types of transport systems. Transport can occur through energy-dependent and energy-independent processes. Several general classes of transport processes have been identified. Passive diffusion occurs spontaneously without the involvement of metabolic energy or of transport protein. It only allows the flow of material down a concentration gradient, and the rate of this process is a linear function of the concentration gradient. The rate of passive diffusion depends on the ability of the perment to dissolve in the membrane biolayer and thus depends on the polarity of the perment and its size. These factors are related to the ability of a perment to fit into transient defects that form in the membrane biolayer. Only water and a few hydrophobic molecules use this mechanism for entry into bacteria. Facilitated diffusion requires the operation of a membrane protein for passage of the perment across the membrane. These transporters merely provide a route for diffusion of their substrate down its concentration gradient, and thus the concentration of the substrate on both sides of the membrane will become equal. Transport is not dependent on the polarity of the substrate and usually exhibits stereospecificity in which isomeric forms of the same compound are transported at very different rates. Because of the involvement of the transporter as a catalyst for a movement, the rate of transport can be saturated in the same manner as an enzyme-catalyst reaction. Good day, this is Professor Will, CBMD, PhD. Today's Wednesday, November 20, 2019. It is 8:44, 17 seconds Eastern Standard Time. And this concludes my short, concise reading on the functions of the cytoplasmic membrane. Well, I hope you enjoyed listening. Have a great day.